Okay, so welcome back to this next video in which we are discussing phospholipase C enzymes. Okay, right. So we're now going to talk about phospholipase C zeta. Okay, and there is only one phospholipase C zeta enzyme, which is just called phospholipase C zeta. Okay, right. So firstly, let me tell you about its expression profile. So it has a very, very limited expression profile. It's only been found so far in sperm cells. Okay, and it's believed to be extremely important in fertilization. Okay, so let me show you now the structure of the phospholipase C zeta enzyme. Okay, so here is the C terminal. I think I'll work backwards for a change. Okay, right. So then what you have is the C2 domain. Okay, and then after the C2 domain, of course, well, upstream of the C2 domain, of course, you then have the Y box, which makes up part of the tin barrel-like domain. Okay, and again, you have the same structures as always. So here is that XY linker, which is obstructing the catalytic site here. Okay, and then prior to the X box, what you do have again is 4 EF hand domains. But the strange thing here with phospholipase C zeta is that it's the only one that doesn't have that N-terminal plaque-string homology domain. Okay, and this is going to hold true when we look at the final phospholipase C eta family. They do have the plaque-string homology domain. So phospholipase C zeta is the only phospholipase C enzyme that doesn't have that N-terminal plaque-string homology domain. Okay, but it does have these other core domains, the EF hand domains, the X and the Y box clearly. If you don't have that, you don't have the catalytic domain. Okay, the XY linker, which is again performing this auto-inhibitory role, and then after that the C2 domain. Okay, right, so as I say, this is found in sperm cells, and it's believed to play an extremely important role in fertilization. Okay, so let me just remind you of the basics of fertilization, and then we'll see what the phospholipase C zeta is believed to be involved in the process of fertilization. Okay, so let me draw a sperm cell here. Okay, so there's its tail. Okay, here's its head, and we'll have the nucleus of the sperm cell shown here. Okay, and then remember, around the nucleus, kind of like a helmet, you have the acrosome, which is basically just a large vesicle, a massive, great secretory vesicle full of enzymes, which are going to help the sperm to fertilize the egg cell. So this structure ahead of the nucleus here, this is known as the acrosome, and it's full of enzymes. Okay, right, now let's draw the egg cell, and this will be completely out of proportion. Okay, in reality, the egg cell is a giant compared to the sperm. The sperm is like a little speck compared to the egg cell. Okay, so here's the egg cell, here's its nucleus here. Okay, now remember, the egg cell isn't just on its own in the fallopian tube, okay? It's surrounded firstly by a structure known as the zona pellucida, which is basically like a gloop of glycoproteins, similar to mucus, surrounding the egg. Okay, and that's something that the sperm cell is going to have to get through if it wants to fertilize the egg. So here is the zona pellucida, which we will just abbreviate from now on down to the ZP. And also, the egg cell actually has a bunch of minions surrounding it. It has a bunch of much smaller cells surrounding it, which it brought from the ovary. Okay, so these are cells from the ovary that were originally around the egg cell in the ovary and which have come with the egg cell. Okay, and you'll have more than just one layer of these. They'll make up multiple layers. Okay, and these cells are called follicular cells, and I won't draw them all the way around because it will take too long, but in reality, the egg cell is coated with these follicular cells. Okay, so in the fallopian tube then you've got the egg cell with its zona pellucida and with its coat of follicular cells floating along. Okay, right, so if a sperm cell wants to fertilize the egg cell, the first thing it has to do is weave in between the follicular cells to get down to the zona pellucida. So that's the first thing that will occur. So I'll just put that in, so weave through the follicular cells. Okay, now it will be at the zona pellucida. What then happens is a component of the zona pellucida, specifically a protein called zona pellucida 3, combined to a receptor on the surface of the sperm cell, and what this triggers is the sperm cell to release the enzymes stored in the acrosome onto the zona pellucida. Okay, and this has a very special name. Okay, this release of the enzymes within the acrosome onto the zona pellucida is known as the acrosome reaction. Okay, so if I just draw this on my picture here, so we'll have a sperm cell here basically, and I'll have to draw it quite big so that we can actually see it, but in reality it will be a lot smaller than these follicular cells. Okay, so here is the tail, like so. Then we've got the nucleus here, and now what's going to be happening is this acrosome is going to start fusing with the membrane basically and releasing its contents. So just like you get vesicle exocytosis elsewhere, so for instance at nerve terminals, this vesicle will be fusing with the cell membrane just like that, and releasing the enzymatic contents onto the zona pellucida. Okay, and that release of the contents of the acrosome onto the zona pellucida is called the acrosome reaction. Okay, and this will break down the zona pellucida basically, and then what can happen is through the sperm whipping its tail, so through the whiplash action of the sperm's tail, it can get through, squash its way through the sides of this hole that it's created and get down to the egg cell, and then what will happen is the sperm cell membrane will fuse with the egg cell membrane. Okay, so if I just show this fusion event, so here is the sperm with its tail here, now fusing with the egg cell here, and of course the nucleus from the sperm cell will go and meet the female nucleus, and they will, after the female nucleus has gone through the second meiotic division, which remember it still hasn't until fertilization occurs, they will fuse after that. Okay, so this is fusion. Okay, now one of the first things that happens in fertilization is something called the cortical reaction. Okay, now let me tell you what the cortical reaction is. Basically, the moment the sperm fuses with the egg cell, you start getting these calcium waves propagating along the oocyte. Okay, so basically this involves a propagating rise in calcium. So this is what a calcium wave means. Calcium goes up here, okay, followed by calcium going up here, followed by calcium going up here, followed by calcium going up here. So you get a rise in calcium that swoops across the cell membrane, across the cell cytoplasm. Okay, and then calcium will go down in all of these areas, and then it will happen again. So you basically get wave after wave of calcium, where calcium goes up, 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 and the propagation of the release of calcium from the ER. So the calcium is coming from the ER in this case. Okay, so what you get is calcium waves being produced. Okay, and these calcium waves cause the exocytosis of loads of little vesicles that are in the cortical portion of the oocyte, okay, which are called the cortical vesicles. Now these cortical vesicles contain enzymes which are involved in transforming the zona pellucida proteins. Okay, so these are called the cortical vesicles. Okay, so the calcium waves that are propagating across the oocyte will cause the release of these cortical vesicles. The cortical vesicles will release enzymes into the zona pellucida, and these enzymes modify the zona pellucida, and they turn it from a sort of snotty gloop into something that's very solid. Okay, so it goes from being mucousy to being solid, okay, and forms a solid crust around the oocyte, and this prevents any more sperm fertilizing the egg cell, any more sperm getting through, and therefore it prevents polyspermy. Okay, right, so this cortical reaction is extremely important for preventing polyspermy. And what causes the cortical reaction? Well, it's this wave, these waves of calcium that are propagating across the cytoplasm of the cell. Now, what causes these calcium waves? Well, as I say, the calcium for the calcium waves is released from the ER, okay, the endoplasmic reticulum of the oocyte. Okay, how do we release calcium from the ER? Well, one of the main ways is by generating IP3. How do we generate IP3? The breakdown of PIP2. Okay, so it's thought that it's the phospholipase C zeta that is actually breaking down the PIP2 in the oocyte membrane, creating the IP3, and the IP3 is then driving this release from of calcium from the ER, which causes these calcium waves, basically these propagations in the level of calcium across the cytoplasm of the cell. Okay, so the thoughts are that the phospholipase C zeta in the sperm cell goes into the oocyte cytoplasm when the sperm cell fuses with the oocyte, basically. Okay, so not only do you deliver the male nucleus, but also you deliver phospholipase C zeta, and this triggers the calcium waves. Okay, so that's where phospholipase C zeta is believed to be extremely important in physiology. Okay, right. So now let's move on to our final family of phospholipase C enzymes. Okay, and this is the phospholipase C eta family, and very little is known about the phospholipase C eta family. It's the one that's been most recently discovered. Okay, right. So at present we don't really know whether phospholipase C eta enzymes are expressed, but there are two of them, phospholipase C eta 1 and phospholipase C eta 2. But as I say, the expression profiles of these are not particularly well characterized at the moment. Okay, now they have the sort of canonical domain structure for phospholipase C enzymes, just like the phospholipase C delta enzymes, in fact. So you have the amino terminus here, then you have the plextrin homology domain here, okay, the N-terminal plextrin homology domain, then our 4 EF-hand domains here, so these are our EF-hands, followed then by our catalytic domain, this tin barrel-like domain, made up of the X box and the Y box, linked by that XY linker, which performs the auto-inhibitory role, okay, here. And then after the Y box, what you'll then have is the C2 domain here, and then the protein ends with the C terminus here. So all the normal structures, just like the phospholipase C delta enzymes, in fact. And again, just like the phospholipase C delta enzymes, the plextrin homology domain is actually believed to be important in the phospholipase C eta enzymes. So this is actually believed to bind membrane phospholipids and therefore bind the phospholipase C eta enzymes to the underside of the phospholipid bilayer. But again, just like the phospholipase C delta enzymes, this only binds you to the underside of the phospholipid bilayer. You're not yet activated. You're not in an orientation, well, the tin barrel domain isn't in a orientation relative to the membrane that will cause the XY linker to come out of the catalytic site. Okay, so at the moment the XY linker is still in the catalytic site, and therefore although the phospholipase C eta enzyme can bind to the membrane on its own, it's not activated. So what does activate the phospholipase C eta enzymes? Well, it's believed to be G-beta gamma complexes. Okay, and again that means that in order to actually activate these, you need to activate the GI/naught cascades. Okay, because remember, those are the ones that have enough heterotrimeric G proteins to actually produce the sort of level of G-beta gamma concentration that's capable of having an effect. Okay, so GI/naught cascades are particularly effective because you have so many GI/naught heterotrimeric G proteins, and therefore you can produce very large concentrations of G-beta gamma. Okay, and the beta gamma complexes of heterotrimeric G proteins are believed to bind to the phospholipase C eta enzymes that are bound to the membrane by binding to the catalytic site here. Okay, so they bind to the tin barrel-like domain here, and then they reorient the tin barrel domain at the underside of the plasma membrane, such that, again, it becomes nicely adjunct to the underside of the phospholipid bilayer here, and that then causes the electrostatic repulsion between the negative charge of the phospholipids in the inner leaflet of the phospholipid bilayer and the negative charge of the XY linker, causing the XY linker to then come out of the catalytic site, and then you've got the phospholipase C eta active. Okay, but as I say, very little is known about the phospholipase C eta enzymes, but we think that they're probably activated downstream of GI/naught cascades, which produce these G-beta gamma complexes. Okay, right, so that now concludes our discussion of the phospholipase C enzymes.